And so from this experimentation, we come to understand the central dogma of molecular biology. This is the general flow of genetic information. Know this flow. When DNA is made into more DNA, we call that process replication. That DNA is typically converted to RNA, leaving the nucleus, in a process called transcription. And that RNA then becomes translated into proteins. It is the proteins that are the doers, movers, and shakers of the cell. Now, there are specialized enzymes that can allow the reverse of information, reverse transcription, if you will. But generally speaking, the flow is DNA to RNA to protein. Let's talk a little bit about replication, the synthesis of DNA into more DNA. It starts with double-stranded DNA. It ends with double-stranded DNA. Transcription then takes the double-stranded DNA and synthesizes a single-stranded RNA message that can leave the nucleus, go out into the cell, and be expressed. Spread the news. Translation is what happens to that messenger RNA, and it is the synthesis of a polypeptide proteins chain that is able to do stuff, to actualize the instructions. And so recognize the specialization. DNA stores genetic information. RNA sends messages, translates it, and proteins do the work. So now that we knew DNA was the genetic material, we can examine it more for its structure and utility. Know the hallmarks of DNA, which stands for deoxyribonucleic acid. The DNA molecule consists of two polynucleotide strands that spiral twisted around each other. We call this a double helix. These two strands are anti-parallel, meaning they run in opposite directions, and they are held together by hydrogen bonds. The nucleotides come in two forms, purines and pyrimidines. The purines are double-ringed, and the pyrimidines are single-ringed. Chargaff's studies on DNA structure, specifically the anti-parallel nature of DNA, gave rise to two particular rules. The first one is that the base composition of DNA will vary between species. The percent of T's versus A's versus G's versus C's would be unique to humans or monkeys or mice, and that all organisms within that species would be relatively similar. But between species, there would be distinct differences. Chargaff's second rule was that the number of A's would always equal the number of T's, and the number of C's always equal to the number of G's. We would be consistently pairing a purine base with a pyrimidine base. And this made sense. Because if you could pair any two nucleotides together, how could you have a template strand? And then, given the size differences between a purine and a pyrimidine, your DNA might be 4 rings wide or only 2 rings wide. That inconsistency would make it liable for damage, nicks, improper packing. So by consistently pairing adenine to thymine, cytosine to guanine, we were consistently getting a purine and pyrimidine pair. So the DNA helix was always 3 rings wide. It was recognized early on that replication would involve copying information from the parent strand to create new daughter strand or newly synthesized DNA, suggesting some sort of complementary or template mechanism to fill in the gaps. There were several hypothesized models. A conservative model would suggest that the parents separated, served as templates, and then rejoined. A semi-conservative model would have this parent strands separate and each serve as a template for the new strands, which would be composed of both old and new. And a dispersive, which would chop up the DNA into a number of segments and copy different pieces. So each resulting daughter strand would be a mix of old and new for many generations to come. Messelson Stahl conducted their famous experiment on DNA replication using E. coli bacteria as a model system. And they set out to determine which of those models was the model. And so they hypothesized by using parent DNA strands that were tagged blue and daughter DNA strands that were tagged red, they would be able to discern conservative, semi-conservative, or dispersive patterns based upon the resulting color of subsequent DNA copies. What they found was in the first generation, there was a blue strand indicating that the parent DNA had been made correctly. Replication after that, it was a purple strand. And in the third replication, they had purple and red, suggesting that the second copy was a mix of blue and red. And after that, all subsequent copies contained only the new red nucleotide. It begins at an origin of replication. This is where the two DNA strands are separated, creating what's known as a replication bubble. A eukaryotic chromosome may have hundreds of these, and replication will proceed in both directions until the entire molecule is copied.
DNA replication begins at specific chromosomal sites called replication origins. At the origin, the DNA duplex is melted by specialized proteins, resulting in a replication bubble. DNA synthesis starts at the origin and proceeds bidirectionally. Two replication forks move outward in opposite directions. At each growing replication fork, two new DNA strands are made. The leading strand is synthesized continuously from a single RNA primer. It grows in the 5' to 3' direction, the direction of replication fork movement. The lagging strand is copied discontinuously from multiple primers, resulting in a succession of Okazaki fragments. Although the direction of DNA synthesis is opposite replication fork movement, the overall direction of lagging strand growth is toward the replication fork. As replication proceeds, the RNA primer of the first Okazaki fragment is replaced by DNA. Then the lagging strand fragments are ligated together.
So at the end of each replication bubble, a replication fork, a Y-shaped region is formed. This is where the new DNA strands will be elongating. Enzymes such as helicase, topoisomerase will assist. The helicase untwists and separates the DNA double helix. Topoisomerase corrects overwinding at the other side of the replication fork. Once open, single-stranded binding proteins will bind and stabilize the single-stranded DNA so they don't collapse in and reseal. Next in comes primase, an enzyme that can start an RNA chain from scratch. It will lay down a small runway of RNA nucleotides using the DNA template. This is known as the primer. It is the starting point for DNA elongation. DNA polymerase can attach to the primer and add nucleotides. It can move really fast, roughly 500 nucleotides per second in bacteria and 50 per second in human cells. Finally, the enzyme ligase is used to join any two fragments of DNA together, where these origin of replications fuse into one. At the replication fork, DNA elongation is happening in two directions, creating two different daughter strands. One is known as the leading strand, the other is known as the lagging strand, because it's being elongated in small segments that will then need the additional step of ligase to join them together. When describing DNA, we coin one strand the leading strand and the other strand the lagging strand. Let's see this more in detail.
In this image, I labeled the top original strand 3' to 5'. I labeled this bottom original strand 5' to 3'. That's the original DNA that is going to be replicated. DNA is unwinding here thanks to helicase. In this example, it will keep unwinding in this direction. Primase places primers. DNA polymerase is building the new strands. Now, the thing about DNA polymerase is when it's building a new strand, it can only build the new strand. in the 5' to 3' direction, meaning it adds new bases to the 3' end on the new strand. See how it's being built in the 5' to 3' direction? This one is called the leading strand. But take a look down here. So DNA polymerase once again is building a new strand in the 5' to 3' direction. But there's a bit of a problem here. See, as DNA unwinds, because DNA polymerase can only build the new strand in the 5' to 3' direction, it has to keep racing up here next to where this unwinding is happening. You can see why then this new strand is known as the lagging strand. On this lagging strand, primers have to keep being placed in order for DNA polymerase to build. These fragments that result are known as Okazaki fragments. Primers have to get replaced with DNA bases since the primers were made of RNA. Ligase, the gluing enzyme, as I like to nickname it, has to take care of the gaps between Okazaki fragments, sealing them together. At the end of replicating, you have two identical double helix DNA molecules from your one original double helix DNA molecule. We call it semi-conservative because the two copies each contain one old original strand and one newly made one.
In terms of visualizing this process, we often describe the replication complex as moving down the strand, a train on the train tracks of our DNA, when in reality, it's a stationary complex that reels in the DNA. And while we focus on a single replication fork, it's important to point out that in a eukaryotic chromosome in particular, where the genome is large, the big solution to the time size limitation is having multiple origins of replication, many of these replication bubbles that will ultimately fuse together. This middleman is ribonucleic acid, or RNA. The monomers for RNA are very similar to DNA. They're nucleotides: adenine, cytosine, guanine. The big difference here, though, is we use uracil instead of thymine. Another interesting thing is the type of sugar. DNA has a deoxygenated ribose, and RNA has an oxygenated ribose. Also, to the strands. DNA is naturally double-stranded, while RNA is single-stranded. Be aware of these key differences. Transcription is sometimes referred to as RNA synthesis. It's the process of making snippets of information from the DNA template. There are 3 types of RNA, and each one has its own specialized function. mRNA is the messenger RNA. This is used to communicate and pass genetic information from DNA to protein. rRNA, the R stands for ribosomal, and this type of RNA can fold upon itself and, like its ribozyme ancestors, engage in enzymatic processes. This actually forms the ribosome, which participates in translation. tRNA, the T stands for transfer. Like the ribozyme ancestors, this RNA sequence will fold upon itself and become an enzyme doer that participates in translation by moving the amino acid to the appropriate nucleotide codon. The process of transcription can be broken down into 3 simple stages: initiation, elongation, termination. Transcription begins at a sequence known as the TATA box. The TATA box is a specialized area of the genetic code that's rich with adenine and thymine nucleotides. You might recall that these nucleotides bond together with only 2 hydrogen bonds, as opposed to cytosine and guanine that bond with 3. This means that that segment is easier to separate, making it more accessible to be read. There are enzymes and other proteins known as transcription factors that will bind to this promoter region to help stabilize the DNA and unwind it to make it more accessible. But the key player here is RNA polymerase. It synthesizes the RNA by following along the DNA strand and adding the complementary base pairs. So here we can see that in visual diagram. We can see our TATA-rich promoter box and the transcription factors binding to that region. And just where the start point is we see our bubble separate those two strands of DNA and an RNA transcript being formed, moving along in the 5' to 3' reading frame. This brings us to elongation. The RNA polymerase will move along the DNA, going 5' to 3', untwisting the DNA helix, making 10 to 20 bases accessible at a time. And it will add a complementary base based upon the DNA template at a rate of about 40 nucleotides per second in eukaryotes, creating the single-stranded, newly made RNA transcript. How does it know it's done? In bacteria, polymerase will stop at the end of a terminator sequence, and the mRNA is ready to go right then and there. It's released and able to be translated or folded up and utilized as a tRNA or rRNA immediately. In eukaryotes, however, the RNA polymerase will continue for hundreds of nucleotides past the termination sequence until it reaches a secondary encode. In eukaryotes, it needs to have confirmation, a double end, are you sure you're sure stop code, before the pre-mRNA is cleaved from the growing RNA chain. I call it a pre-messenger RNA because it's not yet ready to be translated or fold up and act as an RNA, ribosomal or transfer RNA. It needs to have some modifications. Translation is often referred to as protein synthesis. This is the process of reading the mRNA message and translating the nucleotides into corresponding amino acids to create proteins that can act as transporters, communication signals, enzymes, or provide structural integrity and strength. The key player of translation is the ribosome. The small subunit of the ribosome will bind to the mRNA binding site, recognizing the start codon. Large subunit of the ribosome will bind on top of this, sandwiching the mRNA transcript. Another key player is tRNA, the transfer RNA. Each tRNA carries a specific amino acid and has a specific anti-codon on the other end. And so when the units of the ribosome come together, the transfer RNA can bind complementary to the RNA transcript and deliver the appropriate amino acid. Once it does, the tRNA will leave, and an amino acyl tRNA synthetase enzyme will help match it back to its correct amino acid so it can be reused and redeliver new information. The 3 stages of translation are very similar. There's an initiation phase, an elongation phase, and termination phase. All 3 stages require protein factors that will aid in the process. Translation begins when the small ribosome subunit and its special initiating tRNA find a start codon, a special unique nucleotide sequence of AUG. Once they sandwich, additional proteins called initiation factors will help bring the large subunit to complete this complex. During elongation, the process proceeds in a 5' to 3' direction. Binding sites within the ribosome, an A, P, or E site, regulate the addition of amino acids one by one. This involves elongation factors, codon recognition with complementary pairing, a peptide bond being formed, and then the shifting of the ribosome, translocation down the transcript. In each of those sites, something unique is happening. The A site stands for attachment. This is where the amino acyl tRNA will bind to deliver the appropriate amino acid. It has to match the appropriate codon and anticodon pair. The P site is where the peptide bond will form, delivering and depositing the correct amino acid. The E site is when the tRNA, now without its amino acid, will leave to come back again after it has been fused with a amino acyl tRNA synthetase. Know what's happening in each site. A, it attaches. Then there's translocation. The ribosome shifts, and what was in A suddenly is now in P. And in the P spot, it will peptide bond to the growing chain. And then there's translocation. What was in P now moves to the E site, but now it's lacking its amino acid, and so it's kicked. E for exit. This will continue until a stop codon in the mRNA reaches the A site. At this point in time, a release factor, which is really a water molecule, is added. But because this is not held together by the same bonds, it stimulates the release and freeing of the polypeptide chain. Part of the efficiency of translation comes from the fact that any number of ribosomes can be translating a single message RNA simultaneously. This enables the cell to make many copies very quickly. So here we can see it in both graphical representation as well as in an electron scanning micrograph.
When the RNA copy is complete, it snakes out into the outer part of the cell. Then, in a dazzling display of choreography, all the components of a molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. Each transfer molecule carries a 3-letter code that is matched with the RNA in the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecules. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second. And as a result, our muscles, brain, and all the vital organs in our body receive the oxygen they need.